Hello, this is Yashveen. In this video, I'll be solving paper 2 variant 2, May June 2019 9700 biology. 1. Figure 1.1 is a photomicrograph of a low power image of a part of the common sunflower Helenthus anus. Figure 1.1 is a transverse section. A state with reason whether figure 1.1 shows a section through the root or the stem of the plant. So this would be the stem because you've got vascular bundles, these structures around the edges of the image. B. Cell type A in figure 1.1 has a large central vacuole. Suggest with reason the role of the tissue formed by this type of cell. So the cells present in the center of the stem would provide support, which allows the plant to stay upright, and that support is provided by the internal pressure built by the volume of water inside the vacuole of these cells, so that the pressure pushes the cell walls outwards, helping the plant to stay upright. C. Structure B in figure 1.1 contains phloem sieve tube and xylem vessels and other tissues that provide support. 1. Name the structure B. Again, those structures are vascular bundles. 2. The actual length of the structure B is 650 micrometers. State the actual length of the structure B in millimeters. So you divide this with 1000, and so this would be 0.65 millimeters. D. When the structure B is observed at a higher magnification, more detail of xylem vessel elements and phloem sieve tube elements can be seen. Outline the differences in the structure of a xylem vessel element and a phloem sieve tube element. So the xylem vessel transports water and minerals, and phloem sieve tube is specialized to transport assimilates, and so they would have difference in their structures. So xylem has no cytoplasm or really any organelle, whereas phloem does. It does have rough endoplasmic reticulum and cytoplasm. Xylem vessel does not have any sieve plates. Unlike phloem sieve tubes, xylem also has pits for lateral movement of water, which phloem sieve tube does not. And so you can describe any differences between the two. 2. Some tissues of the gas exchange system include cells that are able to produce and secrete mucins. Mucins are stored in vesicles in these cells, ready for secretion. Once outside the cell, mucins absorb water to form mucus. A. Name the structures in the gas exchange system that produce and secrete mucins. So those would be goblet cells and mucus glands. B. Mucins are described as glycosylated proteins. The process of glycosylation involves the addition of sugar components after polypeptides are synthesized. Suggest one location in the cell where glycosylation of mucin could occur. So processing of polypeptides or proteins can occur in either rough endoplasmic reticulum, where there are ribosomes attached to it, or in the Golgi body. C. The processes that occur in the production and secretion of mucins are listed. Complete table 2.1 by writing the processes in the correct order in which they would take place. So you've got transcription and translation. The first stage is part of the first stage, which is the production, and then you've got glycosylation, which is the processing of protein, and then it would be secreted by exocytosis. So we start off with transcription followed by translation. Dechlorine ions move out of the mucin producing cells at the same time as mucin is secreted. Suggest and explain how the exit of chlorine ions helps the formation of mucus from mucin. The production of mucus from mucin involves water movement into mucin to form mucus, and if the movement of chloride ions causes this movement of water, that means chloride ions somehow play a role in the movement of water by osmosis, which is caused due to a water potential gradient. When chloride ions move out of the cell, the water potential outside of the cell decreases, as a result of which water moves out of the cell and enters mucin to form mucus. E. The gas exchange system includes some cells that are able to divide by mitosis. Explain why it is important to have these cells in the gas exchange system, or in other words, the significance of mitosis in gas exchange system. Generally, mitosis is required for producing genetically identical daughter cells so that they can replace dead cells and repair damaged tissues. 3. Figure 3.1 is a photomicrograph of human blood cells from a healthy individual who lives at sea level. The cells labeled C, D, and E are white blood cells. A name cells C, D, and E. The smallest would be the lymphocyte, so that would be D. And between C and D, the one which has larger nucleus is a monocyte, and the other one is the neutrophil. B. In humans, an increase in the white blood cell count can be associated with leukemias and with infectious diseases such as measles. Chronic lymphocytic leukemia is a type of cancer that starts in the bone marrow, and in early stages, many people with chronic lymphocytic leukemia feel well. The disease is sometimes diagnosed by chance during a routine blood analysis when a high white blood cell count is noticed. Many of these white blood cells are only partially mature. One suggests why chronic lymphocytic leukemia starts in the bone marrow and not in any other location in the body. That is so because leukemia, or chronic lymphocytic leukemia, is a cancer of stem cells that are present in the bone marrow, because these stem cells are the cells that would differentiate to form lymphocytes. To explain why a high white blood cell count is a feature of measles and CLL. So measles is an infectious disease, and they cause increase in number of white blood cells because they stimulate immune response, where the number of B or T lymphocytes can increase by clonal expansion following clonal selection. And the number of white blood cells can also increase in chronic lymphocytic leukemia, because leukemia is a type of cancer where the cells divide uncontrolled by mitosis. C. Most of the oxygen that enters the mammalian circulatory system is transported by red blood cells. 1. Describe and explain the passage of oxygen across the cell surface membrane of the red blood cell. So, because oxygen molecule is a hydrophobic molecule, it can pass through the hydrophobic interior of the cell surface membrane by simple diffusion. 2. At a high altitude, the partial pressure of oxygen in the atmosphere is lower than at sea level. If a person travels from low altitude to high altitude and remains there for a few weeks, the red blood cell count increases. Explain why the body needs to respond to high altitude by increasing the number of red blood cells. Because the partial pressure of oxygen is lower in the air, that means the amount of oxygen inhaled would be lower, and therefore the affinity of hemoglobin for oxygen would be lower as well. This would decrease the percentage saturation of hemoglobin with oxygen, and to compensate for lack of oxygen, a greater number of red blood cells are synthesized so that greater amount of hemoglobin is available to bind with the oxygen molecules. D. Polypeptide synthesis occurs before a red blood cell is released into the circulation. The HBB gene codes for the beta polypeptide, beta globin polypeptide of hemoglobin. There are two alleles of HBB known as HBA or HBS. Describe the difference between HBA allele and HBS allele, and state how the difference affects the beta globin polypeptide and the hemoglobin molecule. Since a gene is a length of DNA that codes for the polypeptide, that means the two genes would have difference in the length of DNA. That difference is a nucleotide. So the sequence of nucleotide in the two genes is different, which is caused by a nucleotide substitution in the right HBB gene. And as a result of this, an mRNA is transcribed that codes for the amino acid valine instead of glutamic acid. Originally, glutamic acid in the beta polypeptide was supposed to be present on the surface of the hemoglobin molecule, which is a globular protein. Now, because it's replaced by valine, which is a hydrophobic amino acid, this makes the hemoglobin molecule insoluble. The bacterium Vibrio cholera is the causative organism of the infectious disease cholera. V. cholera has structural features typical of all bacterial cells. It also has a flagellum for movement. A figure 4.1 is an outline drawing of V. cholera. Complete figure 4.1 by drawing and labeling the structures found in it. So you're supposed to be drawing structures that are typical structures of a bacterial cell. You could draw a plasmid, a cell wall, 70 as ribosomes, and cytoplasm. And because this question is of 4 marks, 4 labels would be all you need. Here I've drawn a length of DNA which is not associated with proteins or inside a nucleus. B. WHO collects data about cholera from 194 countries that are members of the World Health Assembly. In 2015, there were cases of cholera in 42 of the member countries of WHO. The total number of cases of cholera reported was 172,454. There were deaths as a result of cholera in 23 of these countries. The total number of deaths from cholera reported was 1304. The case fatality rate for the cholera is the proportion of cases of cholera that results in death within a particular time period. A country with cases of cholera that are properly treated should have a case fatality rate of less than 1%. 1. Calculate the case fatality rate for the 42 member countries of the WHO for 2015 and give your answer to the nearest 0.1%. So fatality rate is the proportion of cases of cholera that result in death. That means the total number of cases that lead to death divided by the total number of cases. And because you're supposed to be giving your answer in percentage, you'd multiply the fraction with 100. So that would mean 1304 divided by 172,454 multiplied by 100. And the answer of that is equal to 0.75, and because we're only supposed to give the answer to the nearest 0.1%, that would be 0.8. Too many of the 23 countries reporting deaths from cholera in 2015 had a case fatality rate of less than 1%. However, 2 of the 23 countries that had fatality rates greater than 5%. Suggest 2 explanations for the higher case fatality rate in these 2 countries. To answer this question, you need to explain using factors that could have prevented death or after infection, that is the treatments. Because here we are dealing with fatality and not just the number of cases. And so that could mean no oral dehydration therapy was available, or the antibiotic course was not completed for the patients of cholera, or maybe antibiotic resistant strains of cholera have caused all of these deaths. In 2010, the country of Haiti experienced a major earthquake. This led to an outbreak of cholera. One, explain why earthquake may lead to cholera outbreak. Here you need to write about the factors that complete the transmission cycle, because outbreak is caused by infection in a great many number of people, and that would only happen if the transmission cycle is completed. And so you could write about how an earthquake is followed by lack of bottled or safe drinking water. The earthquake might have damaged the water treatment plants, so the water could have been contaminated by the pathogen, or about how there is no safe disposal of sewage. Figure 4.2 shows data about cholera collected by WHO over a period of 8 years from 2008 to 2015, where the data includes the total number of cases of cholera for each year, represented by these bars, and the number of countries in each year that had the cases of cholera represented by the graph plotted using these dots. Comment on the trends shown in figure 4.2. So because this question is asking you to comment, you could describe and explain it. I'd start by describing the general comments. So the number of cases is fluctuating over number of years, and the number of countries that had cases is generally decreasing. As for similarities, the greatest number of cases and the greatest number of countries that had cholera were in the year 2011, and the differences would be between in 2008 to 2010, where the number of cases of cholera is increasing and the number of countries with the cases is decreasing. In this case, this increase in 2010 occurs might have occurred because of the earthquake that had occurred in 2010 in this country, Haiti, over here. So you could write about that, and that would cover 3 points for this answer. 5 cells contain carbohydrates, proteins, lipids, and nucleic acids. A figure 5.1 is a list of biological molecules, some of which are components of larger molecules. You've got to complete table 5.1 by using only the molecules listed in figure 5.1. Each example can be written under one or more correct headings. All the examples in figure 5.1 should appear at least once in table 5.1. Starting with cellulose. Now, cellulose fits into the category polysaccharides because it is a carbohydrate molecule made up of many monosaccharides, and it also fits into the category polymer as it's made of many repeating units, which are beta glucose molecules. And thymine nucleotide is a monomer which forms part of the polymer DNA. And then moving on, we've got alpha glucose and beta glucose. Those would be monomers as well as monosaccharides. Monosaccharides because they are single sugar units, and monomers because alpha glucose is part of the starch and glycogen molecules, whereas beta glucose makes the polymer cellulose. And then messenger RNA it's not a monomer, it's not a monosaccharide or a polysaccharide, but it is a polymer. Similarly, glycogen is also a polymer as well as a polysaccharide, since it's made up of the monomers or monosaccharide alpha glucose. And then glycine is an amino acid which makes up the polymer polypeptides, so that glycine would be a monomer, followed by alpha globin, which is a polypeptide, and so it would be a polymer. We explain how the structure of in the phospholipids, the phosphate heads face towards the aqueous environment where they form hydrogen bonds, and the fatty acid tails that are hydrophobic face towards each other and form hydrophobic interactions with each other. C-catalase is an enzyme that catalyzes the breakdown of hydrogen peroxide, which is a waste product of metabolism. The reaction catalyzed by catalase is shown in figure 6.1. So hydrogen peroxide is broken down by the enzyme into water and oxygen molecules. A student carried out 2 experiments to investigate the progress of the reaction shown in figure 6.1, where potato tissue was used as the source of the enzyme. 6 pieces of potato were cut, each measuring 20, 10, and 10 millimeters. In the first experiment, hydrogen peroxide solution was added to 3 pieces of the potato tissue, and the progress of the reaction was measured. Figure 6.1 shows how the first experiment was set up, and part 1 asks you to suggest how the progress of the reaction could be measured. To record the progress, you either measure the rate of disappearance of reactant or rate of appearance of product. Now, because you are given the apparatus to measure the volume of gas over here, you do that, and so you'd measure the rate of appearance of product, which is where oxygen is a part of the products, and so you'd be measuring the rate of appearance of the product. And you do that by measuring the volume of oxygen at recorded time intervals, and then you could plot a graph using that. 2. In the second experiment, the student cut each of the 3 remaining pieces of potato to obtain 6 pieces, each measuring 10 by 10 by 10 millimeters. Using exactly the same conditions, the student measured the progress of the reaction and obtained different results to the first experiment. Explain why the results of the second experiment were different from the results of the first experiment. In the second experiment, due to having a smaller size, the potato pieces have a larger surface area to volume ratio, which means greater amount of active sites of the enzyme in the potato tissue are exposed, which leads to higher rate of formation of enzyme substrate complex, and therefore these in the second experiment the rate of reaction would be higher. B. The student then investigated the effect of temperature on the activity of catalase. On figure 6.1, sketch a curve to show how temperature affects the activity of an enzyme such as catalase. So up to the optimum temperature, as the temperature would increase, the rate of reaction would also increase, and then beyond the optimum temperature, increasing the temperature would cause the enzyme to denature, and therefore the rate of reaction would fall rapidly. And this is it. We are done with this paper. Thank you for watching.